This video is going to demonstrate how to do a hands-on activity for membrane transport. Membrane transport is one of the core concepts in anatomy and physiology. If students can really master the concept of movement of ions across a membrane, then tissues like muscular, nervous, digestive, urinary can be so much more understandable. It goes beyond just memorization. I wanted to create a nice hands-on activity that the students could easily use time and time again. What you'll see down here towards your right is a quick write-up in my lab manual where I describe some background information on homeostasis and membrane transport, both active and passive, and then also a quick procedural steps involved with the activity that we're going to perform in the activity that I'm going to demonstrate today. Now obviously the students will have a textbook that will have more background than that, but that was meant to just kind of set the stage. What my students do is work in groups of 4. And so during the second week of the semester, when we're just reviewing through some chemistry and cell structures, we're going to take one period where they're going to get a large dry erase board. They're going to draw 3 cells lined up side by side as if they were an epithelium, in this case lining the intestines as it were. They will label them according to the apical surface, the basolateral surface. They'll label the relative ion concentrations of sodium and potassium. High sodium on the outside, high potassium on the inside. We're going to demonstrate how the movement of ions move across here. And so I'll use these little bingo chips to represent the sodiums and potassiums that they can easily manipulate in the activity itself. Now, I've also taken a number of these Lego blocks to create what are the membrane proteins. I've tried to color code them. So for example, what you see here is a passive channel that allows potassium to move through. And you can easily swivel it to be a closed channel or an open channel. And so that way the ions can easily move from high to low concentration. Very simple, very graphically, very kinesthetically. We can do the same thing with sodium. We can also take a look at something like this little pivot here. This will serve as an active transporter. This would be the sodium-potassium ATPase. And so when ATP drives this, it's going to be pushing sodiums in one way, potassiums in the other. There are 3 tasks that the students are going to perform during today's lab. The first is simply to review the electrochemical gradient across the membrane. So as we said, high sodium outside, high potassium inside. The 3 pieces we're going to give the students are going to be the potassium leak channel, the sodium leak channel, and the sodium-potassium ATPase. And so the students will take some time figuring out, okay, if we set a potassium leak channel on the membrane, which way is potassium going to move? And then same thing if we do sodium, and they can play with it on either the apical side or basolateral side, and then they can also bring in the sodium potassium ATPase, and they can figure out how you can move these ions across. Simple, straightforward. The second task is now to give them the challenge of maintaining a membrane potential of high sodium outside and high potassium inside. And so to do that, again, they'll run through and they'll figure out how to be able to set up in which direction do they want the pump to work. So they'll talk among themselves. You'll go around each group and ask questions, answer questions, and they'll eventually figure out that they can establish that if they set it up so that the sodiums drives outward, potassiums drive inward, the channels can be closed at times. And so now we're going to be able to easily move our sodium's relatively outside, potassium's relatively inside. The second task of the day now is going to follow upon this, and so now what we want to do is we want to give the students the challenge of we want to move sodium chloride from the lumen of the intestines to the bloodstream. And so let's add in some ions to represent chlorides. And then the players that they will use for the second task are going to be the potassium leak channel, the chloride leak channel, our sodium potassium ATP pump, and a sodium potassium chloride symporter. And so this protein will use the sodium gradient to drive the pump, which will then move sodium, potassium, and chlorides across the membrane in the same direction. So the students are going to take a look at this. They're going to figure out the fact that, okay, if I put the sodium potassium chloride symporter on the apical surface, that's going to help me to use my high sodium concentration to drive potassium and chlorides into the cell. I can put a chloride leak channel down here on the basolateral surface, which will help to continually to drive my chlorides all the way through to the bloodstream. If I add a sodium potassium ATP driven pump on the basolateral surface, that's going to drive my sodiums out, which is going to keep the intracellular concentration low. And then I can put an apical potassium channel, which will help me to take the relative potassiums back out from intracellular to extracellular. And so those can continue to move with the sodiums, helping to drive the chlorides across. So now I've moved sodium and chloride all the way from apical to basolateral. Now granted, I am hands-on paying more attention to the chemical gradient of ions. We know that the electrical gradient is more important, and that's something you can discuss with your students, but this helps to get the general concept in mind. This will take the students quite a bit of time to work on, and that's fine. They'll be demonstrating application and mastery. All right. If you really want to push things along, the third activity we can do is we can kick the game up a bit and say, okay, let's add glucose to the lumen of the lumen of the intestine. And so now I want to be able to move glucose from the apical down here to the basolateral. And so the players for this are going to be a passive glucose carrier, my sodium potassium ATP pump, and a sodium glucose symporter. Now this is a membrane protein that will use a high sodium concentration to help drive glucoses in the same direction across the membrane. So our students start playing with this again. All right, first thing we're going to take a look at is put our sodium glucose symporter here, helping to use sodiums to drive glucose across. I can put my passive glucose carrier down here on the basolateral surface, opened up to allow glucose to be accepted. And then I can put my sodium potassium ATP driven pump on the apical surface, and that's going to help to drive my sodiums back out. Did I say that right? Yes. And so I use my sodiums to help drive the glucose all the way across. You can work with all sorts of options like this. This gives you an idea of what the students can be building. Now we can pick this up again in a couple of weeks when we get to nervous tissue. We could draw a large neuron. We can use some of these very simple, straightforward proteins and help the students to figure out how membrane potential works, action potentials, local gradient potentials, synaptic events, muscle tissue, so on and so forth. Take your time watching the video, read through my lab description. Let me know if you have any questions.